So yeah, my group is a mix of all kinds of different multidisciplinary people, and on our best day, we span the gamut from really very conceptual, almost philosophical kinds of questions to quantitative theory and computer simulations to wet lab experiments that give rise to preclinical and hopefully someday actual biomedical applications. So you can see all of the primary stuff here, the papers, the data sets, the software, everything is here. And then this blog is kind of my own personal ideas on what I think all of these things mean. So let's start today's overview of the multiscale human with this. This is a very famous piece of art called Adam Names the Animals in the Garden of Eden. And there's something very interesting about this, and there's something that's fundamentally wrong and something else that's, I think, extremely important. One interesting thing about this story is that it was up to Adam to actually name the animals. God couldn't do it. The angels couldn't do it. It had to be Adam that names the animals. And what's interesting is that in these ancient traditions, naming something means that you've discovered its true nature. Okay, by giving something a name, or actually by finding out its true name, it means that you've really understood what this is. And I think the thing about this that we're going to have to change is the idea that there is a fixed set of natural kinds that he, we all know what these different animals are, and Adam is a natural kind, and he's in fact different from all of these. So all as I'll show you today, I think all of that has to go. But this idea that we are going to have to do the hard work of understanding the true nature of some very unconventional beings with whom we will have to share our world, that I think is profound and will affect all of us going forward. So today I would like to do three things. I would like to talk about what we are, and then what does this mean for biomedicine? So some of the facts around our multiscale nature. What does that mean for regenerative kinds of applications going forward? And a little bit at the end about what I think this means beyond repair of the standard human anatomy. So one of the most important things that we are in continuity with a lot of fascinating biology of other creatures. So I want to show you what some of your kin are capable of, so that we get an idea of really how plastic and rich biology is beyond what we typically think of as models of health and disease. So this here is a tadpole of the frog Xenopus laevis. You've got the nostrils here. Here's the mouth. Here's the brain, the spinal cord, and the tail. And this is the gut. And you'll notice two interesting things. One is that there are no primary eyes here. We've prevented those from forming, but we did form an eye on the tail. Okay, and when you do that, when the eye forms, it does put out an optic nerve. The optic nerve sometimes synapses onto the spinal cord here, sometimes it goes to the gut, sometimes nowhere at all, but it never goes to the brain. It never makes it all the way to the brain. And yet, when we built this machine to test these animals for behavioral learning in the visual assay, so different light stimuli and so on, we find that they learn perfectly well. They can see. These animals can see out of these eyes. Isn't it remarkable that an animal with a radically different sensory motor architecture, with an eye on its tail that does not connect to the brain, in fact can use it with vision in its behavior? We didn't need extra rounds of evolution, no mutation, no selection, no adaptation needed. This works out of the box. Why does it work out of the box? And I'll show you some of these things. But you start to get the idea that there's some very interesting plasticity here that goes beyond simply making a reliable body every time during embryonic development. Here's another interesting thing. This animal is a planarian, a flatworm. They have a true brain, bilateral symmetry. They're similar to our ancestor, our direct bilaterian ancestor, and they have this one of their fascinating properties is that you can cut them into pieces, and when you do cut them into pieces, every piece regrows the rest of the worm. Every piece knows exactly what a planarian is supposed to look like. So they're highly regenerative, but they're also smart. And you can train them. In this case, for place conditioning, you can feed them on these little bumpy surfaces, and they learn to look for food in this location. And then you can cut their head off, containing their brain. The tail has no behavior. It sits there doing nothing until it grows back a new brain. And when it does grow back a new brain, you find out that it still remembers where to find the food. So not only is the information apparently stored outside the brain, but it's also imprinted onto the new brain so that the correct behavior can take place when the brain is developed. So as information moves throughout the body, we start to think, what will happen in human patients when we deal with degenerative brain disease by, for example, implanting naive stem cells into the brain of a patient with many decades of memories and personality traits and so on? At least in some model systems, we see that actually memories can colonize new hardware that appears. So we don't know if this will work in humans, but it's interesting to watch behavioral memories move around through tissues. It gets even weirder because some creatures like this undergo a metamorphosis where this soft-bodied being that crawls around in a two-dimensional world and it eats leaves has to basically tear up its brain, start from scratch, rebuild a new body and a new brain to become this three-dimensional world dweller that flies and drinks nectar. Now, what has been shown is that if you train this caterpillar to look for leaves in a particular color cue, you will find that the butterfly still remembers this information. Now, it is remarkable that the information persists through basically a completely refactored brain. So how does it stay intact is one question. But an even more profound question is the fact that it's not enough for it to persist, because first of all, butterflies don't care about leaves. And they don't crawl the way that these caterpillars do, they fly. So the exact memories of the caterpillar are actually of no use to the butterfly. What the butterfly can use are the kind of generalized lessons that the caterpillar learned, and then it has to remap that information onto a new body with new different with a completely different controller for its motion and so on. So it has to generalize, it has to remap, and all of this happens on the fly. In other words, during the lifetime of this agent, it becomes just a completely different being with remapped and generalized memories. Now, you might think that this is kind of an unusual case. I mean, we are not insects. We don't metamorphose in this drastic way. But the reality is that none of us, cognitive beings, have access to the past. At any given moment, what you have access to is the engrams, the memory traces that the past has left in your body or brain. So in effect, at any one moment, we are these continuous slices, and at any one moment in this slice, all you have to go on in order to decide what you are, who you are, and what to do next is a reconstruction of your past, the dynamic reconstruction of your past from the memory traces that have been left in your body by a learning process. So you can think about your memories as messages. Who are they from? They're from your past self. And like any other kind of message, they need to be interpreted. You don't just automatically know what they mean, you must interpret them. And not only is this true for this kind of cognitive process that we all undergo, but in fact, on the developmental and evolutionary timescale, you have the same thing. You have lessons learned by an evolutionary lineage. These are compressed and squeezed down into a very small representation, which is an egg. Most creatures don't create copies of themselves. They actually create an egg, which is a very narrow, compressed representation of all the wisdom of past generations. And then the embryo has to deflate this process to reconstruct and to decode what actually was there before. But what's really important, and what I will show you momentarily, is that this process is creative. It doesn't actually just literally build exactly what was there before. It uses the information, but it uses it creatively in new ways. And so you're starting to see this really unusual aspects of the biology that aren't really emphasized in typical biomedical settings. Now, it's important to also remember that whatever properties we have, and most, for example, philosophy of mind talk about the capabilities, the moral responsibilities, and so on of the human mind. But we are, of course, at the center of a continuum. We're a continuum evolutionarily, and we're a continuum developmentally. And in both cases, we develop smoothly from a single cell. Right? So whatever you think we are, we have to have a story of scaling, not of categorical differences, but actually how did that emerge and scale up from our very humble single cell origins. Not only that, as I'll mention towards the end of the talk, but we're actually at the center of two continua, because not only are we sort of spread out according to our history, but now with bioengineering and other tools, we can make changes, and we can make changes both biologically and technologically, and therefore it becomes very difficult to say what an actual human is, because where these different properties start and stop on these continua are not at all trivial to say. So one of the things my group does is to work on a framework that I've developed that tries to allow us to think about all kinds of agents. Okay, so this means the familiar creatures like primates and birds and maybe an octopus and a whale and things like that, but also swarms and engineered new life forms and AIs, whether robotic or purely software, and maybe someday alien kinds of beings. And the idea for this framework is that it has to move experiment forward. It has to give us new capabilities in biomedicine and engineering, and hopefully better ethical frameworks for dealing with these kind of unusual creatures. And so all of that is described here. And of course, I'm not the first person to try for something like this. Here's Rosenbluth, Wiener, and Bigelow trying to understand how passive matter eventually becomes second-order metacognition, such as we have, and so on. And so this is all of our journey. We all start life as a single cell, a little blob of chemistry and physics, and then eventually we become something like this, or maybe even something like this. And I really hate this phrase. People will so often look at mechanisms and say, well, that's just physics. Well, all of us were just physics once. And the one thing we know from developmental biology is that there is no sharp line where a magic lightning flash says you used to be physics, but now you have a true mind, true cognition. There's nothing like that. So the appearance of what we take to be really central to being a human is scaled up from the properties of the biochemistry and the physics of an oocyte, and we need to understand how that process works. To really dig into this, take a look at the blastoderm of an early embryo. So there's maybe at an early stage of, there could be a flat disk of 50,000 cells, and we look at that and we say, there's an embryo. But what are we counting when we say there's one embryo? What is there one of? I mean, there are thousands and thousands of cells. What is there one of? Well, what there's one of is alignment. It's alignment between all the cells, both physically, I mean, they literally get aligned, but also teleologically, they get aligned towards the same journey that they're going to take in anatomical space. They are aligned with respect to their model of themselves and the outside world and the shape that they are building. They're all together collaborating on building one very specific thing. Now, one thing you can do is, and I used to do this with duck embryos as a grad student, you can take a little needle and make some scratches in this blastoderm. And when you do this, it's about 4 to 6 hours before they heal up. But before then, each of these islands does not feel the presence of the other, so it self-organizes into an embryo. And then when it heals up, what you end up with is twins and triplets and things like this. So there's something very interesting here. The tissue that we call one embryo, it could actually be anywhere from 0 to probably half a dozen individuals. How many selves, be that ducks or humans in a blastoderm, is not set by the genetics. It isn't obvious. It's determined by these kinds of individuation processes that take place within this blastoderm, and how many beings are in there is set dynamically. And this, by the way, this question of where do I end and somebody else begins, or the outside world, right? Everybody is somebody else's neighbor here in this kind of blastoderm, has a lot in common with questions of cognitive science in terms of split brain patients and dissociative identity disorders and so on, when there are multiple individuals sharing the same brain hardware. So what we are not only scaled up beings from single cells, but we're also a kind of geometric individuation of a potential, of a medium with pretty large potential for how many beings can come out of it. So that's already kind of a little bit disruptive to our view of ourselves as these monolithic kinds of agents. But at least we're a true unified intelligence, right? I mean, we have a brain, we have a centralized brain. We're not like ants and termite colonies and bees and so on that other people call collective intelligence, right? We're a real unified intelligence. Well, that's more complicated too, because here, for example, is René Descartes. René Descartes loved the pineal gland because in the brain there's only one of them, and he felt that was the ideal place for the unified, centralized human experience to be located. But Descartes didn't have access to good microscopes. If he had good microscopy, he would have looked inside the pineal gland and realized that there isn't actually one of anything. There's tons of cells inside the pineal gland. It's not a monolithic kind of construct. There's tons of cells, and inside each of those cells, there's all this stuff. And so there really isn't one of anything. We are all collective intelligences. We are all made of parts, even those of us with brains. This is the sort of thing we're made of. Now, this happens to be a free-living organism called the lacrimaria, but this just shows you what cells are capable of. This is one cell, okay, and here he is leading its life, hunting in its environment. There is no brain. There are no stem cells. This is just incredible competency at its little single cell kinds of tasks. And so what we as scientists and philosophers owe are stories of scaling. Where do large-scale minds come from? How do they self-assemble from these components, which themselves are active and agential? We are built of an agential medium. We're not passive matter, not even just active matter, but actually an agential medium where all the components have their own goals and competencies. In fact, it goes even below the single cell level. So inside of that lacrimaria are a bunch of biochemical networks. And what we've shown is that if you model these networks, for example, gene regulatory networks or pathways, what you can find is that even the networks themselves, never mind the cells, the nucleus, the signaling factors, never mind any of that, just a few, literally a half dozen chemicals turning each other on and off, are already sufficient for 6 different kinds of learning, including Pavlovian conditioning. The chemical networks inside your cells can learn. And this has massive implications. And here we're working on actually developing tools to train these pathways inside cells for drug conditioning and some other applications in regenerative medicine. So what we are then are a kind of self-assembling multi-scale competency architecture. Every level of organization from the molecular networks up is solving problems in their own space. And as humans, we're reasonably well primed to recognize intelligence in 3-dimensional space. So medium-sized objects moving at medium speeds in 3D, so like dogs and crows and things like that. But we're actually very bad at noticing intelligent behavior in other spaces. For example, the space of all possible gene expression, so transcriptional space, physiological state space, and what we'll talk mostly about today is anatomical morphospace, the space of all possible shapes that you could acquire. Now, I think that if we had, for example, sense organs that looked inwards to, let's say, measure 10 different kinds of parameters of your blood physiology, if you could directly sense the physiological parameters in your bloodstream, I think you would have absolutely no trouble recognizing that your liver and your kidneys are intelligent symbionts that traverse physiological state space. They navigate it every day, dealing with all the things that happen, and they help keep you alive. But we're just not very good at noticing that. So this idea of this intelligent navigation of these spaces is what I'm going to describe next. And the reason I'm going to focus on it is because I think it has massive implications for biomedicine. Now, one of the things that evolution has used in our bodies is an electrical system that spans all of these scales. And just imagine if I told you that with the power of my mind alone, I can make ions move across cell membranes, and I can depolarize up to 1/3 of my body just with the power of my mind alone. Now, you might think that I'm either lying, or maybe this is some sort of kind of a yoga or mind-body medicine. It's some sort of weird biofeedback thing. And the reality is, no, all of us can do this. It's called voluntary motion. When you wake up in the morning, and you have all sorts of very high-level goals, so executive-level kinds of cognitive goals, things about your career and things you're going to do during the day. In order for you to be able to get up and do those things, all of those high-level cognitive goals have to be transduced into the motion of potassium and calcium and other ions across your cell membrane. Your high-level mental content makes the ions dance in the membranes of your cells. And the reason that works is because of an electrical system that basically captures that cognitive activity and transduces it down to the smallest level of biochemistry. It's an amazing system, and that's what we're going to talk about. But let's first take a step back and just ask, where does our anatomy come from, right? So we all make this journey from a single cell egg to some kind of species-specific correct anatomy. So there's our navigation of space. But how do the cells know where to go? Because we all start life as a collection of these blastomeres. Eventually, they make this incredibly complicated thing. In the case of a human, I mean, look at all the organs. They're in the right place, the right size, the right shape relative to each other. Amazing order. Where is this actually specified? You might be tempted to say it's in the DNA, but of course we can read DNA now, and we know it's not any of this that's there. It's a specification of proteins, the tiny molecular hardware that every cell gets to have. It doesn't. The DNA doesn't directly say anything about shape, size, organs, or any of that. So we need to understand how that hardware makes decisions in order to form something like this. Same as this is the outcome of the physiology and the computations that the hardware does, and it's not, the outcome isn't in the DNA any more than the structure of the termite mound or the precise structure of the spider web is in the genome of these animals. It's the result of the activities of all of these active agents. So we want to understand, how did they know what to build? How did they know when to stop? If something is missing, how do we get them to repair, to edit it? And as engineers, we'd also like to ask, what else is possible? Could we get these cells to build something completely different? Or is this the only thing they know how to build? And so I visualize the end game of that whole field as the anatomical compiler. Just imagine someday you'll be able to sit in front of a system, and you'd be able to draw the plant, animal, biobot, organ, whatever you want, at the level of the anatomy, not in terms of any molecular properties, but at the exact anatomy that you actually want. And what this system would do is compile that description down to a set of stimuli that would have to be given to cells to get them to build exactly that. Why do we need it? Well, first of all, that if we had something like this, birth defects, traumatic injury, cancer and aging, degenerative disease, all of this would go away. Okay, all of these issues boil down to our inability to tell a bunch of cells what they should be building. Now, why don't we have something like this? Molecular biology and genetics have been going gangbusters for decades. Why don't we already have this? I mean, we don't have anything remotely like this. Why not? And I think that's because it's very important to understand what this is. This is not some kind of 3D printer. This is not about micromanaging molecular biology states or gene expression or scaffolds or anything like that. It's a communications device. It's supposed to translate your needs as an engineer or a worker in regenerative medicine into the goals of the cells so that they build it. Now, why don't we have something like this? Well, here's where we are today. This is what programming looked like in the 1940s and 50s. In order to program this computer, she had to interact with the hardware, physically rewire the thing, right? And the reason that today, when you're on your laptop and you want to go from Photoshop to Microsoft PowerPoint, you don't get out your soldering iron and start soldering your laptop. Why? Because computer engineers have figured out that if your hardware is good enough, if you make reprogrammable hardware, then amazing things are possible by communicating with it with signals, not by rewiring the hardware, but taking advantage of its various computational competencies. And where biomedicine is today is basically at this stage. All the exciting advances, genomic editing, CRISPR, pathway rewiring, protein engineering, all of these things are about the hardware of life. And so we're very good at manipulating which cells and which molecules talk to which other cells and molecules. We're really a long way away from what we really want, which is control of large-scale form and function. And that's because we've been assuming that what we're dealing with is merely a complex machine. And what it really is an agential material. So in order to think about how do we engineer with different kinds of materials, it's useful to think about an axis of persuadability. So the point of this axis is that different systems go at different places upon this continuum, and the idea is that depending on where you are amenable to different sets of tools. So down here, it's just rewiring. You're not going to convince it or reward it or punish it or anything like that. Then you have some cybernetic systems which are kind of interesting. You can reset the set point, like in your thermostat, and you don't need to rewire the whole system. You just change the set point, and it will keep the temperature. In fact, you don't even need to know how the rest of it works. You just need to know what the encoding is like. And then you have these systems, which are incredibly complex, but they hide all that complexity under an interface of learning. As long as you know what the reward and punishment currency is, you can train them, and they handle all the difficulty underneath of tweaking all the synaptic potentials and everything else. And then of course, you have very, very complex agents that you can convince to do things with cogent reasons. So there are sets of tools, all kinds of hardware things, and then control theory and cybernetics, and then behavior science, and then psychology and psychoanalysis and so on. And so now we can ask a simple question: where do cellular collectives fit on here? I will point out that you can't simply guess, and you can't have philosophical pre-commitments to saying that cells are chemical machines and they have to be here and maybe here. You can't assume any of that. You have to do experiments. You have to test which of these many types of tools are actually amenable. So what I'm going to do next is talk about the kind of cellular intelligence, specifically the ability to reach their goals by different means in novel circumstances that cell collectives are able to exhibit. So the first thing I will point out is that development is very reliable, so it happens correctly most of the time. But the reliability and the emergent complexity, the fact that the final product is more complex than this, that is not why I'm calling it intelligence. It's not about emergent complexity. It's not about reliability. It's about navigation of anatomical space that is very, very plastic. So for example, if we cut early embryos into pieces, you don't get half bodies. You get monozygotic twins, triplets, and so on. And that's because the system can get to where it's going in anatomical space from different starting positions. That's kind of interesting, but it gets better. This is one of my favorite examples. This is a cross-section through the tubule of a kidney in a newt. And you can see there's about 8 to 10 cells that normally form this structure. But one thing you can do is you can create polyploid newts that have extra copies of their chromosomes. And when you do this, the cells get bigger. So when you do this, not only do you still get a normal newt, which is amazing. Apparently, it doesn't matter how many copies of your chromosomes you have, but also the number of cells scales to the cell size. When the cell size gets bigger, fewer cells are able to do exactly the same structure. So that's kind of amazing. But it gets really remarkable when you make these newts very highly polyploid. The cells get so big that one cell bends around itself to give you that same structure. So look at what's happening here. First of all, this is a kind of top-down causation. In the service of this final outcome, the cells are using different molecular mechanisms. They're using different tools to solve a problem they've not seen before, to reach the same outcome. In this case, cell-to-cell communication. In this case, cytoskeletal bending. So they use different molecular tools to get their job done. But more broadly, think about what this means for a newt. This is what I meant by the creative interpretation of your genome, that every creature that's born, it doesn't do exactly what its predecessors did, but it can use the tools that they've left for them. So coming into the world as a newt, what can you be sure of? Well, you can't be sure of how many copies of your DNA you're going to have. You can't be sure of the cell size that you're going to have. From some other work I haven't shown you, it also works if you take away a bunch of cells or add a bunch of cells. So you can't be sure of your cell number. It doesn't matter. Under all those circumstances, you have to be able to get the job done. That is the creative problem solving that these cells are doing. You have to navigate that anatomical space with the tools you have, even when circumstances change. Not just the external environment, but you can't even count on your own parts. And so this is also, that kind of problem solving is also what drives regeneration. So some creatures, like this axolotl, are able to regenerate their limbs, their tails, their eyes, their jaws, portions of the heart and brain, just incredibly regenerative. And what happens here is that if they lose part of the arm, and they're always biting off each other's legs, so they will inevitably lose part of their leg, what will happen is that it will regrow, and it will grow exactly the correct structure, and then it stops. And that's the most amazing thing about regeneration, is that it knows when to stop. So this is basically a homeostatic process. When you're deviated from your correct path in anatomical space, you work really hard to try to get back there. And when you get back there, you know you were back there, so you stop. So that process, any kind of a homeostatic process, needs to remember the set point. It needs to know where it's going in anatomical space. How does it know when it's created the correct shape? And moreover, all of the individual cells need to be working together to achieve this purpose. How do they do that? Well, there's an interesting concept that I call cognitive glue, which is basically the set of policies that bind subunits together into a larger scale collective. We know how this works in behavior. So here's this rat. The rat has learned to associate pressing the lever, get a delicious reward. But notice that the cells at the bottom of the feet touch the lever. The cells in the gut get the delicious reward. No individual cell had both experiences. who owns the associative memory of these two things being associated? It's no individual cell. It's the rat. The collective, known as the rat, is able to keep this associative memory between its different parts. And how does it do that? Because it has an important kind of cognitive glue within its central nervous system. So it has neurons that are bound together. And that allows it to learn and to have goals and to execute behavior to achieve those goals. Now, the way this works is that it has these neurons which have ion channels here that enable them to achieve voltage potentials. Those voltage potentials may or may not propagate to their neighbors through these electrical synapses called gap junctions. And by propagating these electrical signals through that network, it's able to process information. It's able to integrate what's happening across space and time across a large body such as a rat. And it's the commitment of neuroscience that if we just track the physiological activity of these neurons, so here is a zebrafish brain tracked for its electrical activity, then we should be able to decode it. In other words, all of the cognitive content, the memories, the goals, the preferences, everything that is in the mind of this animal should be contained within these electrophysiological signals. And we should be able to do what's called neural decoding to get them out. Well, it turns out that idea, the use of electricity and electrical networks to integrate information across space and time, is incredibly ancient. Neurons did not invent this trick. It actually showed up around the time of bacterial biofilms. So all the cells in your body have ion channels. Most of them have electrical synapses and form networks. And we can actually do the same kind of strategy as the neuroscientists did. And one of my claims is that neuroscience actually isn't about neurons at all. It's about the use of information processing networks to integrate competent subunits into a greater collective, an emergent being with a mind, such as a rat or a frog or a human. And we can do exactly the same kind of strategy. In fact, we can port all of the tools from neuroscience. The tools do not distinguish. Okay, that all of the tools of neuroscience can be used outside the nervous system. So now we can ask, we know what the brain thinks about. Brains think about moving you through three-dimensional space, and then later maybe through linguistic space and things like that. What do the rest of the body cells think about? Well, it turns out they think about shape. From the very earliest moment of fertilization, the cells make networks that think about shape. In order to discover this, we had to develop some tools. First, we developed voltage-sensitive fluorescent dye methods that allow us to read all the electrical activities. So here's an early frog embryo where the cells are sorting out who's going to be left, right, front, back, and so on. And then here are some cells in culture. But now we have the ability to read these electrical states without having to poke cells with electrodes. We can get a map of the whole tissue. We do a lot of computer simulations, starting with the molecular networks that transcribe the different channels, and then tissue-level kinds of bioelectrics, and then eventually symbolic kinds of things where we try to understand pattern completion and so on, memory properties of these circuits and so on. Let me show you what some of these fields actually look like, these patterns. This is a voltage dye time lapse of an early frog embryo putting its face together. Now, instead of the colors, this is grayscale, but it's the same idea. The brightness indicates the voltage level. So what you'll see, there's a lot going on, but here's one frame from that video. And what you're seeing here is that prior to the appearance of the organs and prior to the gene transcription that is necessary to make these organs, you already see, we call this the electric face, you already see, here's where the animal's right eye is going to be, here's where the mouth is going to be, the placodes are out here. The tissue already has a pre-pattern that is going to become the anatomy. Okay, and as I'll show you in a minute, this pre-pattern is essential to forming the face correctly. So we can now read out the plans that this collective intelligence of the body has for making the different organs. And again, the reason I'm calling it a collective intelligence is because we are using the tools of neuroscience to understand how collectives exhibit problem-solving behavior in anatomical space. Instead of three-dimensional space, such as conventional behavior, we're looking at the cellular collective doing behaviors and solving problems in anatomical space. Problems like injury, like having the wrong number of and the wrong size of cells, and so on. So this bioelectrics, we can read it. Actually, one thing that's interesting is that it doesn't just hold individual cells together into an embryo. This cognitive glue is actually multiscale because here, if we poke this one embryo, these others find out about it, and the same thing happens here when you poke this guy within a few seconds. All of these find out about it. It's actually it works across scales to integrate active subunits into larger collectives. And in fact, we've done work showing that groups of embryos are better able to solve problems, for example, exposure to teratogens, than singleton embryos are. So there's a collective intelligence within the tissue, and there is one across multiple animals. Now, not only do we know how to read these patterns, the more important thing is to be able to rewrite them, okay? Because that's the only way we know that the patterns are actually functionally important. So we don't use any applied fields. There are no electrodes, waves, frequencies, radiations, no magnets, nothing like that. What we do is use the tools of neuroscience to control the bioelectric interface that cells are normally using to hack each other, which means we open and close their ion channels on the cell surface, and we open and close these gap junctions to control the topography. And we can do this with drugs, we can do this with optogenetics, all the typical kinds of tools. And so now it's time for me to show you what happens when we do this, right? What actually happens when we rewrite the electric pattern memories of the body. So I only have time to show you a couple of examples, but here are a few of my favorites. So you saw that little eye spot in the electric face picture that I showed you. And so one question that you might ask is, if we introduce that voltage pattern somewhere else in the body, what's going to happen? And so you can do that. We can take some ion channel RNA encoding specific kinds of potassium channels. We inject it into cells that are going to become, in this case, gut. And what you see is that actually those cells are perfectly happy to build an eye. And so in this frog embryo, there's the gut, there's the eye. If you section this eye, you've got lens, retina, optic nerve, all the typical things. And so here we learn a few things. First of all, we learn that the bioelectric pattern is instructive. It actually called up an entire organ. Second, we find out that it's extremely modular. We didn't have to tell it how to build an eye. Frankly, we don't know how to build an eye. There's so many different cell types, so many genes that have to come on and off. We have no idea how to control any of that. What we did find is a high-level subroutine call that says to the tissue, build an eye here. And they take care of all the rest. All the molecular steps are taken care of. The other thing we found out is that actually in the developmental biology textbook, it says that only the cells up here are competent to become eye. And that's true when you prod them with the master eye gene called PAX6. If you do that with PAX6, yes, that's true. Only these cells up here will obey and make an eye. But in fact, if you use a better prompt, such as this bioelectric state, turns out that all the regions of the body are capable of doing it. So when we talk about competency and a lack of it, we have to understand, is that the competency of the material, or is it the competency of us as the engineers who don't understand yet how to communicate our goals to it? And we also found out that actually, if you only inject a few cells here, actually the system knows that there's not enough of them to make an eye, so these cells recruit their neighbors. So all this other stuff up here was never touched by us. It's a kind of a secondary instruction, just the way that ants and termites recruit each other to help them do certain tasks. So all of these are competencies of the material. We did not have to teach them to do that. They already do it. So we've used this kind of thing for a regenerative medicine program having to do with limb repair. So frogs, unlike salamanders, do not regenerate their legs. If they lose a limb, 45 days later, there's nothing. What we did was work out a cocktail of ion channel modifying compounds that immediately trigger this kind of MSX1 positive blastema. Within 45 days, you've got some toes, you've got a toenail, and eventually a very nice leg that is touch sensitive and motile. In our latest experiments, 24 hours of stimulation with this cocktail leads to a year and a half of leg growth. A year and a half. And during that time, we don't touch it at all. No scaffolds, no 3D printing, no talking to the stem cells. Just on that first 24 hours, you communicate that they have to go to the leg building path, not the scarring path, and then you take your hands off the wheel. At this point, I have to do a disclosure. So David Kaplan and I are scientific co-founders of a company called Morphoceuticals, which is now pushing this towards mammals and hopefully someday towards patients. We build these bioreactors, which are a delivery method. It's a wearable bioreactor that delivers the ion channel drug payload to the wound and then protects it while it tries to regenerate. So stay tuned for these kinds of things in the future. I want to skip now from the regenerative kind of thing to the topic of cancer, because cancer is actually very interesting in terms of this question of what are we, and the whole multi-scale human idea. So we have this notion of a cognitive light cone. A cognitive light cone is the size of the largest goal that a system can work towards. Not the size of how far it perceives or how far it can act, but the largest goal that it can work towards. So individual cells have very tiny cognitive light cones, and they're very simple things like keeping the pH or the hunger level down at this particular scale. They have a little bit of memory, a little bit of predictive capacity actually, but everything is about these tiny little single cell goals. But during evolution and during development, when they scale up into these large scale networks, they can actually work on really grandiose goals like building these organs. Their memory goes up both in terms of spatially and temporally, and they can project their competencies, their problem-solving competencies, into new spaces, from physiological space to anatomical space. So here's what's happened. The little tiny goals here, much larger sort of grandiose goals here. I mean, no individual cell knows what a finger is or how many fingers you're supposed to have, but the collective absolutely knows, and they know because if you remove some, they'll fix it and then they'll stop at the right level. That process, the ability to connect into a network that remembers these enormous set points, has a failure mode, of course, like everything else. The failure mode is called cancer. So when cells disconnect from that electrical network, they become, so this is human glioblastoma. They basically roll back to the kind of unicellular lifestyle. The boundary between the self and the world shrinks. So from this enormous self here that they're building, this giant thing, the borders shrink, and now they're just amoebas again. And as far as they're concerned, the rest of the body is just external environment. And so these cancer cells are not actually more selfish; they just have smaller selves. The boundary between self and world is flexible, and it can grow and it can shrink, and it's determined by the size of the goals. Your cognitive light cone is determined by the size of the goals that you are concerned with and actively working towards. And so that kind of weird way of thinking about it actually suggests a therapeutic strategy that we explored, which is that if we inject a human oncogene into these tadpoles, they will eventually make a tumor. If you track the bioelectrics, you can see right away where it's going to be. These cells are electrically disconnecting from the rest of the body and acquiring a depolarized potential, and then you'll get metastasis and so on. So what we did was we said, instead of, we're not going to try to kill the cells, we're not going to try to fix the oncogene. All we're going to do is reconnect them to the electrical network of the rest of the body. And we'll do that by co-injecting an ion channel RNA that basically just keeps them hyperpolarized so that the gap junctions stay open, so that they stay connected. And what you get is this is the same animal here. The oncoprotein is labeled in red, and you can see it's all over the place. It's still here. The cells are not dead. The oncoprotein isn't gone, but there's no tumor. And there's no tumor because it isn't the genetics that drives the outcome. The genetics sets the hardware. The hardware can do many different things. And one of the things that it can do is continue working on this large-scale goal of making nice skin, nice muscle, and all of that. So what we're seeing here is that this question of the multi-scale kind of architecture that we have, and the ability of each of these levels to determine where the boundary of its concern is a really critical thing to understand and manipulate for biomedicine. And we're now moving from these frog models into human or cancer spheroids and so on. Okay, and so the last thing I'll show you, and then I'll stop here, is that so far we've looked at the ability of multiscale cellular systems to reach goals, to do so in surprising circumstances, meaning to solve problems, and to use bioelectrical networks to achieve those goals. One more remaining question: where do these goals come from? And again, we're tempted to say, well, probably evolution. In other words, over the years of the lineage, the specific tractors in all those problem spaces have been fixed by the environment and selection and so on. So I want to show you examples where that kind of thinking breaks down. So I'm going to ask you to think about what this is. So you look at this and you might say, well, it kind of looks like something we got out of a pond somewhere. It looks like a primitive organism. If you wanted to guess the genome, you would probably guess that it's similar to some kind of primitive cellular creature that lives in these aqueous environments. And you wouldn't really know its goals and competencies. But what I can tell you is that if you were to sequence this, what you would get is 100% Homo sapiens DNA. These are human cells. This is made of adult tracheal epithelial cells. If I told you that we were using a human genome, you would not know that it's capable of building this weird autonomously moving biobot. This is not like any phase of human development. They have some interesting capabilities. One thing they can do is traverse these kind of scratch wounds. So this is human neurons, iPS-derived human neurons. We put a big scratch through it. So they go through this scratch, and then they land, and when they land, they form what we call a superbot cluster. So there's probably 8 to 10 or so of these biobots here, and what they do when they form this is they start knitting the two sides of the wound together. If you lift them up 4 days later, you see that what they were doing down here is connecting the two sides of the neuronal scratch. Now, who would have thought that your tracheal epithelial cells that sit quietly in your airway for a long time, when taken out of the body, no new genetic circuits, there's nothing changed about the genome here, no scaffolds, no weird molecular biology, just letting the system reboot its multicellularity into another form gives rise to an autonomous little proto-organism that it actually has a number of amazing properties, but one of them is that it likes to heal neural wounds. So what you can think about is something we call an agential intervention. Someday, you will be able to donate some cells from your own body. Those cells will be made into these Anthrobots, put back into your body to be able to heal wounds, interact with other tissues, drop off pro-regenerative molecules, do something to the microbiome, look for cancer cells, all these different things. Because these things already have their own autonomous activity. They already know what inflammation is. They already, and they come from your own body, by the way, so you don't need immunosuppression. So putting everything together, I'm going to just claim that in the future, this side of the space of biomedicine is going to develop rapidly. Right now, everything is here, focused bottom up, focused on the hardware. Okay, these are all the things that are in wide use today. But there are all these other options. We haven't even talked about our data on training cells and tissues. But here are some biobots made of patient cells, so agential implants, all the different electroceuticals, which are part of a broader class of morphoceuticals, which are basically top-down controllers that don't seek to micromanage symptoms. They don't seek to establish specific biochemical states. They are signaling to the highly competent cellular collective to undergo very complex behaviors that we do not know how to micromanage. So the future in biomedicine, I think, is going to be much more about communication and collaboration, not micromanagement. And I think it's going to look much more like a weird kind of somatic psychiatry, not like chemistry. Right, and we're actually using some AI tools to help us communicate with the rest of the body through the bioelectric interface, but there are many other interfaces as well, of course, biochemical, biomechanical, and so on. But the idea is not to micromanage molecular states; it's to communicate new goals, reset memories, and take advantage of the highly modular capabilities of the cellular collectives. So in the last couple of minutes, I just want to point out what all of this entails. Remember that in the whole first part of the talk, what I emphasized was the plasticity, the idea that cells and tissues cannot assume much of anything about what they should be doing. They have to solve problems on the fly. They're given certain tools in the form of genetically encoded hardware, but after that, they have to creatively form with some kind of functional being, maybe a standard embryo, if possible. If not, it could be an Anthrobot or a Xenobot or any of the other things that we've built in our lab. They have to figure out what they're going to do, and because of that, they are incredibly interoperable. So pretty much anywhere along this continuum, we can add technological components to the naturally evolved ones, right? So you can introduce exogenous DNA and nanomaterials and scaffolds and synthetic organs and all kinds of different things, all the way up through the ecosystem level. Because life is so interoperable, because it does not make assumptions, it will work with whatever it's got. And that means that almost any combination of evolved material, engineered material, and software is some kind of possible agent. So there are cyborgs and hybrids and various different kinds of chimeras and hybrids and so on. Some of this already exists. We already have humans walking around with artificial organs and chips in their brain and so on. This will only expand. and the space of other beings is going to be enormous. When Darwin said endless forms most beautiful, he was very impressed with the diversity of life on Earth. All of that is a tiny little corner of this enormous option space of new bodies and minds. And the interesting thing about all these different creatures that we're going to be sharing our world with is that they are nowhere on the tree of life with us. We cannot judge them in the same way that we're used to thinking about biological lineages. And the old questions that have more or less served us well for years, which is, what are you made of, and how did you get here? Right? Were you made in a factory? Are you made of metal and so on, or are you made, or are you biological? Those things used to be really important binary categories that would determine things like our ethical obligations to you and so on. Those categories are crumbling. I mean, they were never really good to begin with, but they were okay for some years. They're going to be useless in the coming decades. So what we really need to do, and this is a fun word that GPT came up with for me, called synthbiosis, which is the idea that we are going to have to live in some kind of ethical harmony with a wide range of unconventional beings, components of which are synthetic. And we really don't want to be in this position when people talk about it's a machine, or it's a living organism, or it's a human, or it's a robot. When somebody has let's say 50% of their brain has been replaced by various augmentative technologies, you don't want to be this kind of neo-phrenologist trying to figure out, well, is it 51%? Is it those binary categories are just completely useless at this point moving forward. So we are really going to have to develop some new frameworks, and I think the new Garden of Eden picture is going to look a lot more like something like this, where we're going to have to ask, what really is, what really are we? We are not our genes. We are not our standard organs. We're not our standard morphology. We are not our commitment to a life cycle where you get astigmatism and lower back pain and loss of cognitive abilities in your 90s and 70s and 80s and so on. We're not committed to any of that. I don't think that's what a human is. I think we have to start thinking about what makes us truly unique, and I actually think it's the size of our cognitive light cone and the ability to exert compassion, wisdom and compassion in a particular radius across beings, in our environment, and I think that's what makes us humans. It's not any of the really contingent biology that was the result of cosmic rays hitting our material over millennia. So I'll stop here and just summarize. I made the claim that intelligence is basically everywhere, that we really need to learn to rise above our evolutionary firmware and learn to recognize it in very unfamiliar guises. The model system of cellular collective solving problems in anatomical space is kind of a good way to start thinking about really unconventional intelligences, and we're going to have to get much better at this, both for biomedicine and for the ethical flourishing of humanity and other beings. Basically, I try to point out that we are fundamentally a process. We are not an object. We are a self-constructing story, continuously being told by a collective intelligence of multiscale components. And really transformative regenerative medicine of birth defects, of cancer, of injury awaits us to develop tools for communicating and collaborating with this somatic intelligence. And the future is not going to be about crusty old categories of machines and so on. It's about a continuum of agency, about understanding how mind scales from its very lowest kinds of forms up to whatever it's capable of, and likely AI tools as universal translators to talk to these and communicate with these diverse intelligences. So anybody who's interested can look up all the different, these are kind of more popular things that I've written more recently. And I want to thank the people who did all this work. So here are the graduate students and the postdocs that did some of the work that I showed you today, and the undergrads, and our many collaborators, our funders, all the illustrations by Jeremy. Again, disclosures, these are 3 companies that supported some of our work, and most credit of all goes to the model systems, because they're the ones who teach us everything and they do all the heavy lifting. So I'll stop here and thank you.